Sale presentadora. Sale presentadora.
Hello again. Good morning. Now we have had a break. We shall resume with a new session in which we'll analyze the challenges and opportunities of medicines based on monoclonal antibodies. Elena Arroba, Business Development Director of 3P Biopharmaceutical Spain, will guide us as moderator through this discussion, which relies on the collaboration of 3P and Paul Corporation and the participation of Mark Wright, Head of Manufacturing at Nordic Nanovector, Norway. Guillaume Plan, biotechnology, biomanufacturing expert in the Merck Group, France. Also, Sabrina Sensig, team manager, bioprocess specialist for Europe at Paul Corporation, France. And Diego Bochocher, director of strategic development and supply chain at 3P Biopharmaceutical Spain. Over to you.
Thank you, everybody. Good morning. Welcome to this challenging session. Well, first of all, I would like to thank, I'm forced to do it, okay? Thank you, Acebio, thank you, Sodena, for giving us the opportunity to host this session. It's very special for us, for 3P, because we are in our hometown, so being in Pamplona, celebrating this marvelous event after hard times, hard 15, 16 months, almost confined, so it's a real pleasure for us. Today, this morning, this session is going to be a very important one because we are going to talk about monoclonal antibodies. I hope you've heard about this. Monoclonal antibodies are the present and the future of biotechnology. They are diagnosing, they are treating the most important, most critical, and the diseases with the highest incidence, diseases for which either there is not a cure or the therapeutic solutions are not sufficient. The pipeline of molecules in development, either early stage development or clinical stage development, has a very good number of monoclonal antibodies, and the number of products that are approved by the regulatory agencies for commercial supply to patients, they are becoming much more significant with the presence of monoclonal antibodies. This is why new challenges appear. Manufacturing supply is a major issue. And here we are. Here we have a very important panel of experts to let us know which are the challenges we're facing for the development and manufacturing of monoclonal antibodies. So the way we've distributed or created this panel of experts is for each of them to provide their specific vision about this topic. So we have the product supplier vision, the drug developer vision, and the CDMO vision to complement this on a 360 degrees, the opinion and the vision of the main challenges for monoclonal antibodies development and manufacturing. So let's start by introducing our top-level panel of speakers. So representing the vision of supplier of technological solutions, we have Sabrina Sensig. She's a team manager, bioprocess specialist, EMEA at Paul Corporation. Based in Belgium, she brings 10 years of experience as downstream process specialist in the Benelux area. Sabrina graduated from ULB University in Brussels with a PhD and a postdoc experience in molecular biology and immunology. She is supporting the feasibility and optimization study in the downstream purification of biologicals, and her expertise is focused mainly on membrane chromatography and TFF technologies. The vision of a drug development company is provided by Mark Wright. He is the head of manufacturing for biologics at Nordic Nanovector and has over 20 years' experience in biopharmaceutical production and protein conjugation. Working at Nordic Nanovector, he is working hard on their lead product, Betalutin, an innovative radioimmunoconjugate that is facing now lead development stage. Prior to that, Mark was the site lead for Piramal Healthcare's antibody drug conjugate unit and worked on over 20 different ADC GMP manufacturing projects for both clinical and commercial supply. And finally, last but not least, the vision of the CDMO manufacturing of this type of molecules, monoclonal antibodies, is represented by two very well-known companies in the CDMO space. Guillaume Plan is Development and Marketing Manager at Merck Biodevelopment Solutions. Guillaume joined Merck in 2012, bringing over 15 years of biotech experience spanning multiple specialties, including biochemistry, molecular biology, and biomanufacturing. Before joining Merck, he was an entrepreneur and founded his own company, Mitoprod SA, for 7 years. This is a biotech company devoted to the biomanufacturing of RNA. Guillaume received a master's degree in biology and health from the University of Bordeaux and in innovation and entrepreneurship from ESCP Business School. And then we have Diego Boscheuer. He has recently joined 3P Biopharmaceuticals as a new member of the management team of the company as Director of Strategic Development and Supply Chain. Diego is a biologist with molecular genetics and biotech specialization, and he has worked for over 20 years in several companies in the biotech industry, especially in large manufacturing organizations like AGC Biologics, a worldwide leading CDMO, where he operated almost a decade, close to 10 years, both in Europe and in the US facilities that he culminated as Vice President of Manufacturing in Seattle, US. Well, so ready to start. First question I would like to share with you, or first comment, is that considering the current trends that we have in the biotech industry, we're talking about high product demands, strong competition, the implementation of single-use technology also. What is in your strategic position, CDMO, drug developer, product technology supplier, to overcome these challenges? Sabrina, maybe you can start first.
Yeah, I will begin and to give you a little bit my vision as a supplier in this round table. And actually, if you look ahead, PAL is working a lot on the challenges on the maps today. And if you look 5 years ago, the main trend was the continuous bioprocessing. But actually, if you look really now, around the COVID situation do not help, but we are changing a little bit our aspect, I would say, and we are going more and more and on the process intensification more.
Right.
And actually, you will ask me, what does that mean? It's a lot of it's a big word, but actually, the process intensification is focusing on the reduction of waste. Mm-hmm. The waste actually in the production is a big issue today. Mm-hmm. And it's a big issue when you want to upscale it. That's the main trigger. And if we are looking a little bit today on the driver business, what that means? It's the reduction of the cost, is the reduction of the footprint, is easy of use of the device. It's a lot of things, and actually we are focusing more and more as a supplier to work on the reduction of the waste on this different step, I would say, different aspect. And for instance, one example that I wanted to talk today is we can work with the process intensification by combining also all the continuous. It can be completely continuous, but it can be also in a semi-continuous or in a hybrid working process intensification. And one example is one step that we are using a lot in the monoclonal platform is the viral inactivation. Mm-hmm. If we look just only on that step, it takes time, it takes large footprint, and it's 2 ways that we need to reduce on that part today. And actually, we are developing new device to reduce the footprint and to work continuously with different steps just before or just after, because actually the viral inactivation is done normally after the chromatography. But we are working now in a way to implement the viral inactivation step just at the moment of the elution, so during the elution. Mm-hmm. And you see that by combining different steps of the production, you reduce already the footprint because you are working continuously, but you combine 2 steps in one. And you work on the time and on the process labor. Another aspect that we are working a lot also is the production of the buffer. the management of the production of large buffer is important today. We need to point that today, if you produce 2,000 liters of maps, actually it's equal to 20 liters of buffer. Yeah. You see what that means in front of footprint of management. And here we are also, PAL is working also on the buffer management. So we are device where we implement device automation in order to produce on demand at the time where you need it, the buffer that you need at the process step for the purification. And you see here, it's only two examples, but it's many examples that we are working in order to reduce this footprint and to reduce the waste.
Sure. I see Mark doing like this all the time, so I guess you have a lot to say, right?
Yeah, I think from a drug developer's perspective, we have sort of two real differences in strategy. One is for the early phase products, and one of them is really for the later phase. And the later phase, I think that the comments that Sabrina's raising are very relevant. That's where we really start to focus on the sort of process productivity and the optimization, and that's a strong point for us. At the earlier phase, we're more looking for speed. And that's one of the points that quite often, once the decision is made on a project, it has to move quickly. And we're already looking to start next day sort of thing. So for us, it's very, very quick. And from a production perspective, things that CMOs can do to assist in that is things like the disposable technologies, because we're not looking for bringing in dedicated equipment. We're not looking for complicated cleaning of equipment, and it makes those operations a lot more simplistic if we can really bring a process in and the CMO has designed processing steps around the equipment platform that they have. And then as the innovator, we're really just buying into the disposable elements, so flow paths and other pieces from it. And that's probably some of the key elements for the early part of the process. I think, from our experience, that is being adopted, and we're trying to see if we can get more over to the disposable flow path approach to processing. And we've worked at sort of hybrid solutions, some fixed equipment, some which is disposable and reusable. Mm-hmm. So I think as we move forwards through that, then we look to start driving the efficiencies through. But in the first phase, it's really just how quickly can we get material available for the clinic.
Thank you, Mark. Let's see what our colleagues from the CDMO will have to say.
Okay, thank you. For my perspective and the CDMO perspective, the key will be to make the scale-up easy, right? So as Sabrina mentioned, process intensification is important to make things easier, faster, increasing the throughput, but making the scale-up right is what is keeping our scientists awake at night, right? And this is enhanced with linear scalability. So basically, what I'm trying to say here is like that, so being able to scale up faster from early development to commercial manufacturing is the key. So in order to do that, the CDMO is implementing single-use technologies, right? Single-use bioreactors. But not only the bioreactor is a key point, it's also the whole chain of scale-up from development of the process development in Amber 250 to shaker flask or rocking culture bags or even bioreactors in different scales. So we need to use common scale-up strategies to be able to go fast to the market. This will solve all the challenges we have in terms of capacity and in terms of bringing product to life-saving product to the market. So on the other hand, as a CDMO, of course, for us it's very difficult to implement the use of like 20,000 liter stainless steel bioreactor. This will take a lot of footprint. This will take, potentially we will need to increase ceiling height, and then it's expensive platform. It will take a lot of time. So currently in the market, installing a bioreactor like that will take 1.5 years. Bringing single-use technology, what is called a six-pack, which is 6 times 2,000-liter bioreactors. So we'll give the possibility to scale up, right, and then scale down on the same way. So using the same common parameters. That's the key point from my point of view to solve these challenges of lack of capacity, high demand, and this will increase the speed. So currently, as a CDMO, we are trying to reduce the time, right, from the early development to commercial, or early development to starting a batch in the facility, in engineering batch or GMP batch. And the key is to create this the scale-up process in a way that is the same, from a mini bioreactor in the AMR 250 to shaker flasks to culture bags to bioreactor 50 liter, 200 liter, 500 liters, 2,000 liters, etc. So that's point of view from the CDMO.
So Guillaume, what do you think?
Yeah, of course, I fully agree with what has been said, what Diego said, of course, what Sabrina said. When we consider the facility itself, and of course what Mark said, in a customer perspective, for sure. But what if we consider the market? We currently have 100 monoclonal antibodies on the market, able to cure diseases. And I think it's the best hope to fight cancers. So the monoclonal antibodies have the potential to fight the cancers. And hundreds are currently in development at the preclinical or clinical stage. And based on that, and given the fact that we produce monoclonal antibodies now for 40 years, actually, the Nobel Prize was at the early 80s, and it was about discovering during the 70s the hybridomas, right?
An Argentinian guy.
Oh, God,
Yeah.
Argentina, all the time.
So it's not a new class of molecules, but as drugs, they are new classes of molecules. We develop them as drugs now for 25 years with the Herceptin, Avastin, all of these very nice molecules. So there is another challenge, which is the emergence of biosimilar monoclonal antibodies. And now for 5 years, we have to consider this trend as something really critical. And what does it trigger? It triggers a need for the reduction of the costs of production. And given that we have hundreds of these monoclonal antibodies on the market, the competition is huge. The market is much higher than it was even 10 years ago. And we have to be faster. So being cheaper, being faster is really the challenge. So as you said, implementation of single-use technologies is one way of being faster, even being cheaper. But really the cornerstone of being cheaper is the titer that we get. So at the cell line development level, we need to provide now almost, in average, I would say 6 to 8 grams per liter, thanks to bioreactor management. But the cell line itself has to be really robust and very efficient, which was not the case 15 years ago. So it's really a strong challenge that we have to face based on the dynamics on the market. And I think there is another point that we can discuss based on the dynamics of the market as well, which is and based on the fact that our customers have, they need to be very reactive, so we need to be very flexible. They need to adapt to their situation. Most of these monoclonal antibodies are developed by startup companies, emerging biotech companies. So they are financed thanks to venture capital. So they need this, which is, it's very difficult to produce a monoclonal antibody, a GMP batch, but they need it for yesterday when they need it. Because of this very strong needs in a financial perspective to create value and to reach the next milestone for the development of this drug. And for that reason, the tech transfer between an internal facility and a CDMO, or between a CDMO and another CDMO, is a key trend as well to consider. And this is something that we develop as much as we can at Merck in order to support our customers in that way.
I I don't know if it was Diego, or I think it was Diego mentioning briefly about the problem of the lack of capacity in the world. What do you think the market is doing? How we can solve this lack of capacity? This is a major issue.
Yes, indeed. It's a major issue. So basically, I see two ways to do that. So basically, as I said before, the implementation of single-use technology is a way to solve the capacity issue. Why? Because first of all, it's a platform that is very easy to build up. So we reduce a lot of waste in terms of doing CIP, SIP. So this is giving us the possibility to do faster changeover between batches, between lots, between different products. That's one way. So that this is, of course, is a trend in the market right now. A lot of companies are installing single-use capacity. But on the other hand, some other way to indirectly to increase capacity is to also increase product titers, to create cell lines that are more robust, that are, so by doing that, we can reduce volume, right? And reducing volume will increment the capacity. So, but of course, then other challenges will come, right? High 10 grams per liter cell line, mammalian cell lines, then we need Sabrina and her team to develop protein A membrane systems. So, we need to create other ways to manage the high concentration, the higher volumes. But that's another challenge. Could be in another. I know you have a point of view, Sabrina, about that.
Sabrina, maybe you can elaborate a little bit on this, and then we come back to the lack of capacity.
Yes, actually, I have different view of that. And it's true that today we enlarge the volume of the bioreactor, but we are working a lot on the titer. But higher titer, then it can give you a lot of issue on the contaminant level. And on that part, we are developing a device to overcome that issue. Because if you don't develop on the very early of the process purification, you have a big bottleneck and you are blocked on that part. And actually, we worked now more and more on filters, clarification filters that is assuming very high titer and very high concentration of bioreactor, because we are working now between 20 and 50 million per ml of concentration, And actually, this type of filter can overcome this issue today. It's still also, we are still working also on a manner to reduce the volume. Even if we are working with 2,000 liters or 10,000 liters of bioreactor, we have also device to reduce just at the beginning of the process to reduce the volume. We are now developing device called single-pass TFF, meaning that it's based on the TFF. You concentrate at the very early of the process, and you also increase the titer in order to go faster on the chromatography, the first step chromatography, which is the protein A, which is very expensive, and on that part we need to work on that way. And then there is also other type of device where you incorporate chromatography, like Diego told. It's a type of membrane absorber where it's single use. It's plug and play. You do not need a large footprint on that part. And actually, you are working also on the waste of buffer, of time, and footprint.
Wow, really interesting. Let's go back to the lack of capacity topic, if you don't mind. So, Guillaume, maybe continue with the CDMO perspective. What is the market doing to solve this very important threat?
That's, first of all, lack of capacity means that there is difference between the needs and the capacity of providing
Right.
GMP batches, I would say, of monoclonal. So the good news is that it's easier for us to allocate our slots, and I think it's a good point, first, of course. But process intensification and investments into new capacities is another answer to this situation. I think it's currently there is a lack of capacity, but I'm sure in 3 years there will not be any more lack of capacity because the market and because there is a strong competition will adapt to this situation. And we know that there are a lot of investments of capital expenditure into increasing capacities all around the world for monoclonal antibodies production, mostly in Korea and China, but also in Europe and in North America to some extent. So that's the first answers. But I think so. The difficulty of this situation is for pricing. In a CDMO perspective, it's difficult to be able to price, I would say, wisely when there is a lack of capacity and when we know that this lack of capacity will not anymore be a lack of capacity in 2 or 3 years. We want to keep our customers, but we want to, of course, to make as much money as we can. That's the purpose of a privately owned company, and that's the main difficulty. So we sometimes have to manage in a case by case on this point, which is not easy, and that's the difficulty, I would say. But that's for a CDMO. It's a good problem when there is a lack of capacity. Yeah, it's better than the opposite, which could occur in 3 years, given the amount of investment that we can see on the market.
Sure. So, Mark, what is your vision as drug development company?
So from a drug development perspective, I think there's a more diverse set of products out there now as well. So the monoclonal space obviously has the more traditional naked antibody, but coming from Nordic, our experience is obviously with the radioimmunoconjugates, and they tend to be a lot more potent because they're armed antibodies effectively. So we also find it's quite difficult sometimes to find the right CMOs with the right skill gaps. So we often look for relatively small scale production, and that doesn't always fit with quite a few of the CDMOs out there. They're looking for more bigger volume projects. But I think this is becoming more of a trend, is that the antibody drug conjugates and various different modifications of the antibodies are slightly different, and they do need different skills and handling. So I think that's going to be a trend of just the product mix becomes more complicated. I think the other element from the developer's perspective is that trying to manage capacity in the market is compounded by having to deal with multiple different vendors. So within the supply chain, the complexity of working with 3 or 4 different CMO organizations starts to become very difficult to manage. Oops. Because it's also to do with the capacity of each individual site and trying to balance that to hit an overall timeline. Often when you're being driven by time as being a critical factor in the development, particularly for the early projects. So trying to find that mix is difficult, and I think certainly from our perspective, we're looking for CMOs who offer a more integrated service, so more offerings within their network, or ideally within the same building if possible, just so we don't have to manage things like shipments and all the other things which come with it. So it certainly is difficult at the current time with CMOs are in a higher capacity situation at the moment. And trying to integrate all of those activities is a real challenge. So, yeah, it's probably a good time for CMOs, maybe a little bit difficult for the innovators. Not sure what's setting that off.
What happened? Okay, well, so let's try to adapt our minds now a little bit on a little bit more business-oriented topic. Okay, what do you think, in your vision, are the advantages and disadvantages of in-house manufacturing skills versus outsourcing the manufacturing capabilities? Let's start with Mark, because you are the one to hire or to incorporate the capacities in-house.
A very good question, and I think there's a few different pieces to look at within this, and it depends some of your organizational size. But I think coming from a smaller biotech, we outsource the majority of the work that we do. And I think that there are some advantages and disadvantages to that, and a lot of that is down to selecting the right partner. So trying to find a CMO with the right skill mix for doing particularly things like the development work, it can be quite challenging. And when you're outsourcing pieces of work, depending on how you set that up, that can be quite an expensive way of doing this. So then you have to start looking at whether there's any other potential models with the CMOs where you could potentially buy larger blocks of time rather than doing things in standalone pieces of work. And trying to manage that is difficult. What I've found is that having a balance of some in-house capabilities is good because you can build some internal knowledge within your organization. And there is a big question, I think, when you deal with a CMO is who is the sort of product owner of the knowledge behind it, because when we're going out to CMO, we're obviously looking for their experience and their knowledge, but then also we need to develop some of those parts in-house, and getting the balance right is quite difficult. So certainly I think some in-house capability is useful, but I think you can mitigate a lot of that by finding the right partners for outsourcing the work.
Okay, Guillaume, Diego, I hope you're ready to talk about the advantages of outsourcing.
Well, yes. I mean, of course, as a CDMO and as a service company, we would like to offer the whole chain, all the services. But of course, we also need to become technical experts and bring the technical acumen to the table. And so we need expertise in all areas, which is also a challenge, right? So, because we can offer drug substance development, drug substance scale-up development, drug substance manufacturing, commercial manufacturing, and drug products, and everything, of course, is a service company is the best, but then the challenge is to find the balance between offer everything and become an expert on each of the areas. And the key point from my perspective is to find the right talents, the right people who have the knowledge to provide all of this expertise, because again, as a CDMO, we want to create a partnership with the clients, and we want to offer a service, and we want to be experts and provide all the technical expertise that we need and our clients need. So that's the challenge, but then the key is to bring the right talent to our company, our organization.
Guillaume?
Yeah, I fully agree with what I've heard, and I think the answer to the question is not the same if we consider an emerging biotech company willing to outsource or a larger company. But most of the market even for monoclonal antibodies development is made of emerging biotech companies, and an interesting point to this outsourcing approach is that in most of the industries, if you outsource, you increase your risk, because you lack the control of what has been done to a third party. So it's something that has to be considered very strongly. And I think for CDMO outsourcing, it's completely different. Given the skills, the knowledge, the amount of investments which are required, if you want to be able to provide a GMP batch of a monoclonal antibody, you need to outsource. And you will reduce your risk if you outsource. But as Mark said, you need to find the right partner. Otherwise, it's going to be a nightmare. And it is not, of course, what you want to do if you want to develop your own company. So that's one difficulty, finding the right partner. But that's a way to reduce the risk. Of course, it's another point in the outsourcing approach is that most of the time, when you outsource in most of the industries, you increase or you decrease your margins because in the value chain, you have one more person involved in the production. I think it's completely different in our industry because of this exactly the same situation. If you outsource, you don't need to hire skilled people with the knowledge, the experience. You don't need to qualify your facility, everything. And actually, of course, it's expensive to outsource to a CDMO, but it's much less expensive compared to developing everything internally. So that's the approach for an emerging biotech company. But of course, it's a challenge. You need to find the right partner.
Right.
And I fully agree. Just one word about what Mark said just before about the one-stop shop. You need to find a CDMO which is able, for, in your case, for ADCs to provide the monoclonal antibody, the linker, the active molecule, and put them together, etc. It's really difficult, and that's a challenge that we have identified at Merck, and that we want to overcome, is to act as a one-stop shop. We have all of these capabilities and put them together in order to provide one only offer to a given customer. Yeah.
Sabrina, I guess your position is quite neutral on whether outsourcing or integrating the capacities, or maybe it's not the case.
Actually, what can I say when I'm working with both of them?
Yeah.
So I'm working with the developer, but also the partner. It's what I see when I'm going to the field, I would say, is actually the most important thing is working with scalable device. And actually, the partner has more facility to work with them. Actually, the developer can work at low scale, and it can be difficult then when you want to transfer the project to the partner. So I think that the key point here is working with scalable and to have in mind at the very beginning of the development, scalability is the key point for me.
Okay. Well, thank you. Thank you very much for these interesting topics. As we have only 5 minutes left, let's give the opportunity to the audience. Maybe there are any questions from you that you would like to share with this top-level panel of speakers? No raising hands. Okay. Okay, so as we have 5 minutes, there is one topic that I would like that we maybe spend some minutes on. Today is the day of the COVID. I think that most of the round sessions or topics that we've been discussing all along the morning, the topic of COVID has appeared at a certain moment. So let's talk a little bit about it from the drug development or CDMO perspective. Well, it's a matter of fact, okay, there is a problem of capacity, as we've seen also in COVID vaccine manufacturing, and also we are facing some problems in the raw material supply, and the market and life is beyond COVID. I mean, there are other diseases, there are clinical trials waiting for the material to be available. So what do you think that can be done in this scenario? Okay, because difficulties are appearing, and we have to share some ideas with the audience just to give them hope about how to live with these COVID times versus real life problems and real diseases. What do you think about the situation? Silence.
No, I mean, it is very challenging right now, as you said, Elena. It's very difficult to face because we all want to have vaccine available on all levels, but there are some products on the market waiting to be released. There are patients waiting for receiving clinical trials, or even the priority, COVID priority is also taking priority against commercial processes. So I think that the suppliers are doing the best they can to supply the market, but the best thing they can do is to build more capacity and use an approach. I don't think the COVID manufacturers, COVID vaccine manufacturers are using an approach to consider the ways to save, the way they are doing processing, so we can actually get some other supplies to the other market. But I think the best way to deal with this right now is to build more capacity for all the supply chain. From media production to Single-use bag production, the single-use bag is also a big challenge right now. So, and this is what is happening, and I hope in the near future this will be solved.
Okay, Guillaume. As Merck, CDMO, Merck, raw material supplier, how do you see the situation?
I think, you know, that's something that most of the people know, but in Chinese, crisis and opportunity, it's the same word. That's what I've heard. I don't speak Chinese, but that's what I've heard. And I think that's the case, actually. For our industry, I think the COVID situation is behind us. It's still difficult, but we understand that it will go more and more easier in the future. But we have had to face a lot of difficulties, mostly based on supply chain, indeed. But when we consider the growth of the needs on the market for very basic raw materials, it's not surprising. It's you know, it's not only for there was something like 100 projects for vaccine development against the COVID. I don't know how many drug development against the COVID, but based on that, of course, the industry was not ready to face that. But it was an opportunity to improve the supply chain, I would say the prediction of when are we able to provide this raw material to this place, etc. And I think the best is in the front of us and not behind, because most of the suppliers, and I'm sure my colleagues from Paul will agree, we have adapted our processes internally in order to be even better than we were before the COVID crisis in providing the raw materials to our customers.
Sabrina, what do you think?
Yeah, actually, I'm supporting what Guillaume is saying. It's actually, it's true to say that today we are saying to customer, you need to anticipate. And I think that we will need to do that for up to 2022, I think. And we need to be clear that there's still the bottleneck of the raw material, and even if we are expanding the manufacturing site, I think that this year we expand 50% of the manufacturing, and it's still not there, you know, and we are still facing issue on the demands. The demands are there, and the people are afraid about that, but I think that in the 6 coming months, there is something that will turn and it will go better in a point of view of supplier.
And Mark, how is Nordinal Vector suffering, or I hope not suffering, this situation?
It's, as everyone, it's been challenging. But I think one of our key learnings is that a complicated supply chain is very vulnerable to this type of scenario. So for us, that's been one of our big things that we're looking at, is really trying to bring more activities together. So our CMOs are managing a lot of the materials issues, and that's been their challenge to try and get those in. And in our case, it's been pretty successful. But we have a product which has multiple manufacturing steps and a short-lived radioisotope. So it's pretty challenging to try and move that around the world during the middle of the pandemic. But with a lot of hard work, we've managed to do it. But I think, as a general rule, the more things which you can pull together in these circumstances really pays off when these types of events happen. And as we know, you can never predict what's around the corner, but these things will happen. So for us, I think it's a key driver to try and simplify that supply chain down as much as possible.
Okay. Well, so I think that with this last topic, we can finalize our session. Thank you very much for sharing the time with us today. Thank you, all of you, for being here today. And, well, I hope you enjoy a really fruitful day at Biospain. Thank you.
Thank you.
Thanks.
Thank you.
Bye-bye.
Thank you very much for showing us the importance and the impact on health and society of medicines based on monoclonal antibodies. We'll now continue on our journey and hear about the challenges that preclinical research faces in Spain in order to become attractive to the pharmaceutical industry. This session, made possible thanks to the support of Pharma Industria, will be in Spanish. So now, en castellano, ya, pasaremos a hacer esta mesa. A continuación, eso sí, pediremos unos segundos para poder hacer correctamente, tal y como exigen las medidas de seguridad actuales, ese cambio de micrófono. Así que en apenas unos minutos regresamos. Muchísimas gracias. Aquí, por aquí.